This is a production of Cornell University.
So I wanted to thank my students. And this is what our group meetings looked like on the left side of the screen. It was either cubby holes or meeting in Central Park when the situation seems to alleviate before the next variant showed up. But also, particularly, I wanted to highlight the individuals here who contributed to the research. Kimberly Gibson, Sarah Doley, Jorge Berni, and then Stephanie Zullo are students of mine. They are now off to better things. They have graduated. Viviana Medina is a student of Matthew Gilbert in bean physiology. And Santos Barrera, who also contributed a few of the slides, was a long-time researcher at CIAT, and he then recently obtained his PhD at the University of Nebraska in a collaboration. So I will, this is kind of a brief overview of my program. Skip that. I would like to outline my talk is that, and it's well timed because the ICCP or IPCC came with that latest report, and it's as troublesome as ever, and we don't seem to be doing anything about it. So I would like to talk about, briefly introduce the topic, the need to increase and speed up germplasm utilization. And I think that a lot of people are doing this, but the target seems to be ever more complex. I'll introduce then the model I'm talking about. This is inspired by research on domestication hotspot, Phaseolus. There are 7 domestications. I'll explain this to you. And then 2 of these domesticates are tepary bean and lima bean. And they may help us in this dire situation. And then I'll end with a summary. So it's clear that we have been putting out CO2 and other greenhouse gases, and not slowing at a rate that is not slowing down. And you see this in the left part. But also, we have benefited, and this is the domestication work that shows, from a relatively stable climate for the last 10,000 years. And except then right at the end, you see an ever increasing temperature. And we have to deal with that. The issue will not go away even if we were to stop right now all the CO2 emissions and other greenhouse gases. Because you see the difference here. I think I also talked to Zoom. The 2 curves here, this is the emissions. If we take the best case scenario where we stop all the emissions as fast as possible, the light blue line here, the CO2 content of the atmosphere is barely going to decrease. So we are set with a continually heated climate all over the earth. And we see the consequences also. For a long time, people have said, well, where is this global climate change? And as recently, I think there was an article in the New York Times that questioned that. To stay out of US politics, the French president said, you have never told us that we were going to be faced with these catastrophes and so on, upon which the French media put together a clip where they listed almost several instances dating back to the 1970s, where they said, hey, we are faced with global climate change and certainly global heating. Okay. So to give a recent example here, at the left, this was the situation in California in September of last year, not very long ago. This is the situation now in March, and we see a radical change in terms of the drought status. I would say a improvement of the drought status in the state. And it's not finished. They are expecting another atmospheric river this week, and it's going to affect mostly Central and Southern California. Okay, we can say that this is a consequence of global warming, the heating of the oceans. So how do we deal with this heating and this irregular precipitation? Okay. We are going to be faced with drought. And just to simplify, because it's going to come back later on, there are at least two types, major types of drought. The one in the middle, intermittent drought, where you have irregular rainfall. And so the crops have to be faced with sufficient water, insufficient water in succession. And then at the bottom, you see the terminal drought, where the crop is grown as a consequence or based on the residual moisture in the soil. And so when we talk about drought, we have to at least define these two big classes of drought. And I will talk mostly about the two, the intermittent drought and terminal drought. I should point out that this is from a work by Fisher, who was talking about rice. Okay, so this is an issue that goes across crops also. Now, the second theme is that we have these gene banks. They have been established all over the world. USDA has an important germplasm system. I would say that it's probably the biggest and if not the most successful one. But one limitation is the use of it, or the lack of thereof, and also the speed at which we use these accessions in a gene bank. And so I will focus primarily here on this aspect here, the germplasm. How do we get it into the recurrent breeding cycles? Now, I've been working on Phaseolus for quite some time, starting at the gene bank, in Seat, Colombia. And the predictions here is, you see here, this is a work of Ramirez Cabal, and he shows the optimum versus less optimum areas in principally, I would say here, in South America and in Africa. And I highlight these regions because they are so important in the nutrition. Local people. If people haven't eaten beans, they probably think they haven't eaten at all. And it's beans and rice, beans and maize, and so on. Now, if we look at the predictions for the future in the Americas and in Africa, you see that the suitable area, which is the green area, is shrinking with the current materials and in the current way of growing these crops. So this is an incentive actually to look what can we do when faced with this heat, increased heat, and also the irregularity of the precipitation. Okay? So the genus Phaseolus itself has 5 domesticated species. They have been known for some time. You have common bean, which is by far the most important one. You have runner bean, which is a closest or close relative. You have the so-called year bean, which is the least known of the 5 domesticates. predominantly in Central America. But you also then have these two other species, tepary bean and lima bean. What we have found is that two of those species have been domesticated twice. Common bean in Mexico and in the southern Andes, and lima bean, again in Mexico, and in the Andes, but in a very restricted area on the western slope of the Andes in Ecuador and northern Peru. And interestingly, that area also has these wild common bean, but those were not domesticated. And there is an altitude difference. Lima bean is on the lower altitude, and common bean is at a higher altitude. And so it's almost like people made a choice, and they domesticated lima bean rather than common bean in that area. Now, there are differences. Why is it that people domesticated these different species? What you see on the screen is the distribution of the wild ancestor. From the left, you see common bean has the broadest distribution. Lima bean is close. Also, it involves this migration in both species from the area of origin of the genus to Central America, and then most importantly, to the Andes. And there are several breakpoints that are naturally determined. Lima bean has pretty much the same distribution, but it's in terms of genetic relatedness, it's somewhat different. Then you have runner bean. You have the tepary bean, which is concentrated in, you could say, the northwest quadrant of Mexico and also the southwest of the US, principally Arizona. And then the year bean has a very narrow distribution, the wild ancestor in Guatemala. And this is probably because of the fact that it's a hybrid, a stabilized hybrid between runner bean and common bean. Okay. So these are the three gene pools in common bean, and the red one and the blue one have been domesticated, not the yellow one. So these are, again, the 7 domestications. You see at the top row is the wild relative, very small seeds, camouflaged. And then the next row are the domesticated seeds, and you see a tremendous change in size, in color, and so on. Now, another thing that differentiates them is their adaptation. The most important species here, common bean, and I say economically at this stage, is growing in an intermediate environment. Not too hot, not too cold, not too dry, not too wet. On the left of it are the species that are adapted to a cooler, more humid environment, which on the face of it might not be useful in a scenario of global climate change, but who knows? If we want to plant earlier to adapt these species to global warming, maybe we'll have to plant earlier into a cooler season. So this is not that they are not useful, but for now, we don't concentrate on them. On the right, you have lima bean and tepary bean, and they are adapted to drier and hotter climates. It's definitely the case for tepary bean. For lima bean, this is a species with the broadest adaptation in Mexico. Another aspect that we have studied is the domestication syndrome. What are the morphological differences and physiological differences between wild and domesticated types in common bean mainly? And I emphasize these traits because they are still useful today. This is not a matter of the past 10,000 years ago. They affect the productivity and the quality of these crops even today. You have a number of characters here for seed and pod colors and weight. Knowing the color is extremely important in terms of consumer acceptability. If it's not the right color, people will not accept it. And I'm not just talking about red or yellow. No, it's a particular shade of red or a particular shade of yellow. So the consumer, especially in the Americas, less so in Africa, are extremely, how would I say, recognizing, picky, if I could say, about the color of these bees. The first actually character of domestication that we identified, the gene for domestication was the determinacy, which creates a bush bee. And it is actually the gene responsible is a homologue of Arabidopsis. And it has 3 copies in the genome, in the bean genome. And one of them is where most of the mutations, the loss of function mutations for these gene were identified. It is also, we took care of the opportunity in order to demonstrate that it was really the gene. We transformed Arabidopsis with a functional allele of the homologue in beans, actually. And we were able to restore the wild type phenotype in Arabidopsis. But so this gene is important in the bush production of green beans because it shortens the flowering time and making harvest easier. Okay? Photoperiod sensitivity, you will see, is tremendously important, especially in terms of germplasm utilization. The genus Phaseolus is a semi-tropical genus adapted to short days or long nights. And when we plant them in temperate areas, many of these accessions don't flower. So it affects the utilization, the screening of germplasm as well. And there are 2 genes that have been identified based on the materials that we had distributed, but to Jim Weller in Australia and Marta Santalla in Spain. Seed dormancy is also important, and it has to do with the permeability of the seed coat and the composition in terms of waxes. And then more recently, Travis Parker, who is a student and now postdoc and now researcher at Davis, studied the development of the pod during domestication. So a first step was to eliminate or reduce pod shattering. Again, this is a trait that is still important today. And he also then studied the absence of threads or strings, people call, we talk about string beans. So the modern green beans don't have a string. And he was able to figure out what the locus is. It is, again, a homologue of Arabidopsis, in the Hisson gene. At that locus, it is the stringless beans have a duplication combined with an insertion of a retrotransposon in between the 2 copies. That creates an increased expression and it eliminates the string. Now, at the same time, this duplication creates an instability. Through unequal recombination, you can revert at a high frequency, about 1 in 1,000, you can revert to the stringy state. And so we are still looking at how can we deal with this to increase the stability. But it's a major issue in terms of quality control and maintenance of genetic stocks in the green bean industry, the production of varieties. And they spend some kind of $50,000, we don't know for sure, trying to guess every year, just because they have to send down people through the fields to rogue the revertants. At the same time, these revertants are excellent isogenic lines because you have the original state and the revertant state in an isogenic state. So we are sequencing them as well. But again, it illustrates how these domestication traits are still important today. And one of the questions is, what is the equivalent of these genes, the homologue, in these other Phaseolus domesticated species? Okay. So I have to introduce tepary bean to you and lima bean. These are the seedlings you see in the field. To the left is a tepary bean seedling. You see here. And this is a lima bean seedling. This is what they look in the field. At the left, you see common bean. At the right is tepary bean. It's a very branched growth habit, cross-trait, hugging the soils. We think that it's part maybe of the adaptation. We see the same thing in common bean in the ecogeographic race Durango for northern Mexico, where it's dry. They also have this cross-trait habit. And also, interestingly, in the Andean domestication race Chile, also has these cross-trait growth habits, presumably because of their dry environment. There is also a difference in the earliness. To the left, you see common bean, it's still green. The dry patches are these mature senesced tepary beans. And then to the right here is lima bean. So there are differences in average cycle. Clearly, tepary bean is the shortest, has the shortest cycle, which is part of that tolerance to terminal drought. You have to run your cycle as quickly as possible to escape the droughts at the end of the cycle. This is what the seeds look like. The scale here is centimeters. These are photos of the gene bank at CIAT, which systematically takes photos of the seeds and the pods of their holdings. And you see overall, the seeds are fairly small. Again, that is an issue with the consumers. Okay. And there are gene banks. You have the USDA has holdings, the CIAT, ILRI in Africa as a potential forage. And you see that branched growth habit here, but also Australia and Embrapa in Brazil. Most of the wild types, the orange dots there, distributed, like I said, in the northwest quadrant of Mexico, but also spilling over into Arizona. And then the domesticated types in purple have a slightly broader distribution, but not that broad. Okay? And then there are differences among the wild populations in terms of their adaptation to drought. You have types here near at the stream edges, but then going up the slope, and their leaves also change. The ones that are up the slopes are in dry conditions, and they have smaller leaves, so smaller evaporative surface. So this was a collaborative trial with CIAT and Nebraska. Carlos Urea, who is the bean breeder in the Panhandle in western Nebraska, and myself, and Jorge Berni is a student here in Davis, and Santos Barrera is the person from CIAT, who was kind of central to these comparative experiments. And then heat stress trials here were conducted then both in California. The university has this really unappreciated series of experiment stations kind of north, south along the state. And they have very diverse environment. One here is at the so-called Desert Research and Extension Center near the border of Mexico. in the summer, it's, to quote, stinking hot. And that's what we want. And we had done a preliminary experiment growing common bean, lima bean, and tepary bean. Common bean was killed. And the two species that survived were lima bean and tepary bean. Okay, so we had done a preliminary experiment, but then they also conducted experiments in Colombia, the northern coast near the Caribbean, very hot and humid this time. And then Alvarado in one of the valleys of the Magdalena River, which can also be very hot. So the results of this analysis, then, if you compare UC Davis and another station, the West Side Research and Extension Center, the three species here, common bean, tepary bean, and lima bean, are color-coded. So you see the green ones are common beans, and this graph then shows well irrigated and drought. And you see a fairly good correlation, but common bean is the least adapted to this situation. Then comes lima bean in red, and then in blue is tepary bean. And clearly, there are tremendous marked species differences, as well as intraspecific differences in their yields. I will highlight here these TEP varieties. These are actually bred lines. These are not just germplasm land races. And TEP is actually a product led by Tim Porch. Some of you may know Tim Porch. He's a graduate of Cornell. Okay, so it shows on one hand that yes, we can use the land races, but breeding will add to the tolerance of this species. There has been almost no breeding in that species. So MET stands for multi-environment trials. And you see here an analysis just of tepary bean under heat stress and under drought stress. And the two combine a fairly high correlation. But again, these TEP materials come out on top here. So there is room for breeding in this species. And the same thing here, where you look at the yield, which the improved lines figure here as the highest yielders in this situation. Now, one of the things that is interesting here is that, at each of these locations, we grow them under drought, under well-watered conditions. And you might ask, well, how do they group these locations and these treatments? You could say, well, all the drought-stressed materials will group on one side and the well-watered on the other side, or is it going to be grouped according to location? And what turns out is that they group rather based on location rather than treatment within location. To me, it suggests that we don't completely know what is adaptation. We treat them here in terms of, broadly speaking, the general temperature, the general rainfall, or the amount of water supplied during the experiment, but that is not enough. There must be other characters involved in adaptation besides these few simple ones here. I don't have an answer to that question. What is it that really differentiates these locations? What you see here is, this is UC Davis. This is Alvarado in Colombia, high temperature. This is Palmira, also in Colombia, drought and irrigated. Okay, so you see here the pattern that the different treatments tend to group together. It's really separated by location. It's not a new observation from breeding, but it raises that question of we need to know more about it. Now, tepary bean has been of interest to people in common bean because they see it as a source of genetic diversity, and particularly resistance to common bean, resistant to common bacterial blight in common bean. And that has been transferred, to show you, and people have developed over about, I would say, 20, 25 years from near absence of resistance in common bean to fairly high levels of resistance by combining different sources, including the resistance in Acutifolius, in tepary bean. And the intention is then to combine this also with tolerance to heat and tolerance to drought. Whether that's going to be possible depends. I think the common bacterial blight resistance was, in essence, an easy case. It's a major gene trait in tepary bean, although it's more complex in common bean. But they were able to do it using acrobatics, which I will explain. With drought tolerance, we almost showed that it's a genetically more complex trait. Now, the point I would like to make here, the emphasis is always transferring genetic diversity from tepary bean to common bean. But what about the opposite? Let's take this adaptation to heat and drought in tepary bean and build tepary bean as another crop. And I think people start turning around to that, but the emphasis has always still been improving common bean. My point in this talk is, yes, we should be able to do both. And even if the situation persists, really improve tepary bean as a source, as a grain base. So tepary bean here is a domesticated, that has domesticated and wild types. It can be crossed with extreme difficulty. Okay, and people have to use embryo culture and so on. Lunatus actually cannot be crossed to vulgaris and the other species. That has been determined. So this is, I would say, gene pool 4. While tepary bean is gene pool 3, reciprocally between common bean and tepary bean. So that's an issue to take into account. We have done whole genome sequencing of land races, breeding materials in common bean, including these genetic stocks that are resistant. What you see here, the organization of genetic diversity. So you have the 11 chromosomes from 1 to 11. The blue, so the blue color represents an Andean origin of the chromosome, the part of the chromosome. And so these block of lines here from A and D to NABE 14, these are typically Andean domesticates. On the other hand, the red or pink lines from ALB to VAC6 are typically Mesoamerican domesticates. And you see that there is some introgression. It may be asymmetric, but you have more introgression from Mesoamerica to the Andean group, and that is because of breeding, wanting to introduce disease resistance. Nevertheless, we see a few other interspecific introgressions, and the one from tepary bean here is typically at the end of chromosomes. I don't know exactly why, but we have noticed that disease resistance clusters are usually at the end of chromosomes. And that may be because there is higher recombination there. And the recombination creates genetic diversity to face which could face the variability of the pathogen. But it's also maybe easier to introgress a material in a high recombination region than in a low recombination region. That clearly is a caveat in terms of the future, in terms of introgressing these different traits. Okay, so it says we need to have larger populations, maybe to identify rare recombinants. So the contribution here of Santos, who, although he got his degree at Nebraska, he came 2 summers to Davis to help to also screen these materials. What he did was to cross an interspecific inbred line, the INB 834, with a wild relative of tepary bee, the Parvifolius, which has been relabeled recently, but it's, let's say, Parvifolius. So you have vulgaris, acotifolius, and Parvifolius, so that you call these the VAP lines. And to give you the comparison, if you cross vulgaris by acotifolius, you have to make tons of crosses. You see the labels to the left here of the failed crosses. If, on the other hand, you cross this line to Parvifolius, you have a relatively high success rate. And this is just a visual comparison. But you get then seeds that will develop. For a normal vulgaris by tepary bean cross, you have to do embryo culture to retrieve anything at all. Okay? So it is easier to cross this vulgaris containing hybrid inbred line with Parvifolius. And you see then here the developing embryo, and you see a plantlet, and eventually a full plant here. Now, this line is still sterile, but if you backcross it to vulgaris, you get a progeny. So if you compare the normal common bee by tepary bean cross, the number of F1s that he obtained in spite of the large number of crosses was maybe 20. In comparison, the cross of these VAP lines by tepary bean, you could obtain a large number, 10-fold increase in the number of progeny. The number of pods was also larger, and in spite of having a lower number of pollinations. So these VAP lines, what they have allowed us to do is to cross vulgaris with tepary bean at a much higher rate than normal. Basically, initially, crossing vulgaris and acotifolius was low viability, low fertility. So somewhere in that genome, there must be segments of Parvifolius that allow increased success rate in the crosses. We don't know what these genes are. It's an empirical observation. Okay? These lines have been sequenced. You see here the reference genome, which is an Andean reference genome. You have an acotifolius genome. This is a Mesoamerican genome. And then you have these VAP lines, these bridge lines, and interspecific vulgaris by acotifolius. And so they looked at the number of Parvifolius segments that were transferred to the progeny in these VAP lines, and you see a much higher frequency here compared to these interspecific vulgaris by and I like to remind you, these VAP lines are also interspecific, but they involve a third species. And so you have a higher rate of transmission in these VAP lines. There is also heterozygosity, and that may be a problem in maintenance. Okay, so you see the comparison. So this again is the sequencing. The light blue is common bean genome, but then you have introgressions, the purple and the brown here, from Acutifolius, and so large segments. So still there is an issue of recombination, but these VAP lines have an increased introgression of Acutifolius or Parvifolius segments in the chromosome. So Santos then wanted to test out if it made any difference in terms of drought tolerance. And so by crossing these VAP lines with tepary as a source of drought tolerance, you get a hybrid which you backcross to Acutifolius. This is what it looks like. Then backcross it to vulgaris. Another backcross. This is what it looks like. And you see the pods here. There is no evidence, really, marked evidence of sterility. Okay. So eventually he did then test the progeny, and you have these interspecific lines that result from the cross between VAP lines and Acutifolius, and then controls. You have the VAP lines themselves. You have common bean lines. You have these interspecific vulgaris by tepary lines. I think I'm running out of time. Is that? Yeah, okay. And so let me show you the results of this. He then performed drought and heat stress trials. And what you see is that the progeny lines, you can identify this in the squares. You see progeny lines that have better yields, more significantly higher yields under drought in 2 years, actually. Okay. So his interpretation is that we are successful in transferring some of that drought tolerance into lines of common bean that themselves are already more tolerant to drought. And what the field you see here, the comparison between, for example, the original vulgaris by Acutifolius, and this is the process involving this 3-way cross with Parvifolius. Okay. I'll skip a few. Our conclusion is from other studies is that Acutifolius is for terminal resistance, terminal drought, and lima bean is actually for intermittent drought. We are also working on lima bean, and what you see here is we developed a large-scale recombinant inbred population that was useful in developing a reference genome in lima bean with Colombian researchers, and then mapping several traits. On the left, you have determinacy in the same chromosome location as in common bean. You have 100 seed weight, which shows an additive pattern. Flowering time, also heavily determined by determinacy. And then cyanogenesis, which shows epistatic interaction. And the final thoughts here is that if we want to increase the use of gene banks, we have to be involved in pre-breeding, like the experiments in crossability, but also the testing germplasm conversion, using these domestication traits to introduce them into an adapted for germplasm, which is kind of the opposite way that we're doing it. And finally, I would acknowledge these three people because they are going to take over the bean breeding program, the East Africa program, and also we have a group looking at the physiology and the drought with Matthew Gilbert. And so I can retire happily. Well, I won't be retiring, let me tell you, because I'll be working on that lima bean project with the SCRI, with the USDA, but at least I'm somewhat alleviated. And I'm very grateful for these individuals for continuing the research. And I'm sorry, so I'm running a little bit out of time. So thank you very much. And of course, you know Christine. That was the purpose of the slide.
Thank you. I was wondering if your map lines already showed some kind of agronomically or agriculturally improvements in terms of drought stress, or really just better possibility with the tepary.
I think that the fact that they have viability, fertility, that they survive in the field, we think that it's a big progress. But it's not the final detail. There, that's the pre-breeding. There have to be next steps. Yeah. You have to realize what it was. That's why I showed that slide with all the labels and so on. You know, that you try, and they fall off. So having something that is viable and fertile is already a big progress. Yes.
More about what you're developing tepary as a commercial crop. What are the characteristics you're looking to integrate from common bean? Like, what are the most important characteristics?
There is one gene that is every variety in North America has to have. That is the I gene for immunity, I think, for resistance to bean common mosaic virus. It has reached the stage where people cross elite by elite in common bean in North America. Well, they don't even have to screen for it because the two parents have it and the progeny is homogenious. That definitely is the case. Then the other might be some of the consumer traits, seed size, at least reach something that is like a navy bean or a black bean. We have the colors already, but the seed size could be larger. And others, yeah, so that one. We are looking now at pod shattering. We don't think that it's a problem because it comes from a dry area. But we are looking at the genetic basis of that. So I'm sure that once we start growing, the diseases and pests will find their way to the field. And then it's wide open. Yes.
Broader question. Broad bean insofar as market costs for beans, or is it very specific on what they want?
It's getting narrower and narrower. So they are in competition with other places in the country. So common bean has disappeared. Used to be, but then it's competing with North Dakota, Michigan, and so on. So it's now lima bean, black-eyed pea or cowpea, and garbanzos. And garbanzos get planted in the early winter as a winter crop, whereas the others is as a summer crop. But then in California, they're also in competition with trees, nut trees. And I don't understand that because these are going to need water. And I expect them to start complaining that they don't get enough water. Why would you plant a tree? But that's what they do. So I think the economics is whatever yields or brings in most money per unit area. That's the thing. Oh, yes.
So you're saying that at the beginning, lima bean is more of a water user versus the tepary beans are more of a water saver. Yeah. So could you highlight more on maybe the water user part of lima beans? Is there anything you've been able to learn from the water saver beans, essentially try and Is it just really just the growing season and that's why the difference in the lengths?
Yeah, actually, in a sense, tepary bean is not that different from common bean, except for the fact that it accelerates its growth
Okay.
under stress. So it has this plasticity. And when you really, I mean, if you used to walk the fields, you say, hey, I've beaten up on this tepary bean and it grows faster and bigger than
Yeah.
But lima bean, what I think its distinctive trait is that it has lots of biomass, and including a very deep root system. And so under heat stress, the leaf temperature is lower than what you see in common bean or even tepary bean. I think that it is because of evaporation that cools off the canopy. And because of that deep root system. And we have seen the same thing in wild common bean. When you can use wild common bean to improve yield under stress. These are the populations that do that come from dry areas in Mexico, the driest areas in Mexico. And they also have the deepest root system. It's not a strong correlation, but it's 10%. I'm satisfied with 10% because what is adaptation? It's probably a multitude of traits. So there is not one trait that determines adaptation. So 10% is pretty sizable. And so these wild beans, they reshape their root system. If you impose a stress for equal amounts of biomass, they get deeper and less branched root system.
Well, we can ask later, but what legume, do you know of a legume that has a really high level of free amino acids? And what level that? They have the highest digestibility, essentially at 2 to 3 fold more than protein.
I'm sorry. But I can maybe understand why they would have digestibility. They don't have to cleave those proteins.
Right. Yeah. Yeah, I was just kind of.
But the question is, they may also have free non-protein amino acids. Yeah, you know, there's lots of amino acids, but not necessarily the protein amino acids. So are they really nutritional positive or negative factors? Right. It seems like there are some amino acids that, I don't know, I'm trying to go to the depth of my memory.
I'm just wondering if any of these ones just happen to have high free amino acids.
Yeah. I would be careful with that. Yeah, but I won't say anything further. Not to embarrass you.
You mentioned the phenotypes of the stress condition, but you said in one slide you said the phenotype is determined by environment, but not by the stress. So I got confused if you study specific
Yeah.
events.
What I meant to say is that the yield pattern among the standard set of lines is determined, is grouped by location, not by the treatment. And a priori, you could say, well, I'm going to have all the drought treatments on one side and all the well-watered on the other side. No, it's actually said it's grouped by location. And Nebraska seemed to be quite different. We thought, oh, it's a fairly dry environment, going to resemble California maybe. No, it's very separate. So there must be other factors involved. And it's really asking the question to try to find an answer, to draw the attention to people, is that how do we characterize an environment?
This has been a production of Cornell University, on the web at cornell.edu.